Welcome everyone. Today I am going to present you a software called Cytoscape. We can use this program to visualize complex network data. Biological network analysis aims to draw the connection between biological entities as graphs. Interactions can be observed in all levels of biology, from the molecular level to the whole ecosystem level. Thus, using graph theory we can model these interactions as networks between the components of a system. We can use graphs to analyze biological networks such as protein-protein interactions, or ecological relationships, and many more biological interplays. These biological networks, like other type of networks too, are composed of edges and nodes. Nodes represent the entities themselves. For example, in protein-protein interactions, proteins are the nodes, and in ecological networks, the species are the nodes. Edges represent the interaction between the nodes, as we have in our example of proteins. Each edge means a link between the proteins or connection between species. Cytoscape is an open-source software platform for the visualization of complex network data and to enable the easy integration of further attribute data to these networks. You can use Cytoscape to load molecular and genetic network data in several standard formats, and you can project your data with global datasets and functional annotations. You can also use Cytoscape to visualize and examine social network data and perform further calculations with it. It is also possible to use it for general complex network analysis. Cytoscape is a powerful domain-independent visualization and network analysis tool with lots of features and built-in applications. You can download Cytoscape to your desktop for free from the official webpage. After download and installation, we can start to work with Cytoscape. After you open the software, you will see the starting panel. From here you can access sample session files and tutorials. After you load a file, the Start panel will disappear, and you will see the Network View window instead of it. On the top of your window, you can find the menu bar, where you can find the View, Layout and Tools settings, save or load files, and a lot more. Under the menu bar, you can find the toolbar, which contains the icons of the most used functions. The Network panel on the left includes a search bar, where you can get access to several external databases. It also includes a Network Overview panel where you can switch between projects and subprojects. In the bottom right corner, there is the Tab panel where you can examine the exact values of the nodes and edges. You can modify these data too. Although Cytoscape is a versatile tool with several built-in analysis tools and features, using the Cytoscape App Store you can include more databases, features, and analysis types. Researchers and developers from around the globe can develop new apps and give new functions to Cytoscape. Currently, more than 370 apps can be found in the App Store related to Cytoscape, consisting of tools for data visualization, network generation, network analysis, automation, clustering, enrichment analysis, and many more. You just have to open the App Store in your browser, select your favorite app and click the Install button. If Cytoscape is running, the app will be downloaded and installed immediately, and it will be ready to use within a couple of seconds. Now let's check some frequently used apps of Cytoscape. One of the built-in apps of Cytoscape is Index or Network Data Exchange. Index is an open-source platform for organizations and scientists to exchange, share, process, store, and most importantly to publish biological network knowledge. Index aims to bring together biological network knowledge from scientific publications. String is a database of protein-protein interactions based on known and predicted information. You can import the String platform to Cytoscape from the App Store. Installing the app will import all functional associations and physical interactions between protein-protein and protein-chemical pairs from the database. You can do it with just one click. So let's open Cytoscape. You will see the welcome page we discussed a couple of slides before. Now we are going to upload some data. The data is withdrawn from our research group platform, TNMplot.com, where you can analyze gene expression correlations across several malignant and normal tissues. We are going to analyze the MKI67 gene expression correlations in colon cancer patients. On the left, you can see our input table. We have 3 columns, one for the gene names, one for the correlation coefficient, and one for the p-value of the correlation analysis. First, we are going to use only the first column, the gene names. We copy them from the table and will insert them to Cytoscape String app to visualize the network connections of the proteins coded by these genes. In Cytoscape, you can select between queries in the Network panel search bar on the top. Let's select the string protein query. After the selection, paste the gene names to the search bar. You can customize your search using the More Options button next to the search bar. Since the default settings are suitable for us, we are not going to change anything. After that, you can click on the Search button next to the More Options button. This is how the network connection of the proteins looks like in String Protein Database. At the bottom of the table panel, we select the Node Table view where we can get further attributes of our proteins from the string database, like UniProt, string database description, and many more. Now we can import our correlation data to the Node Table. Clicking on the File option in the menu, we can select the Import option and then the Table from File sub-option. In the pop-up window, for Import Data we select option as Node Table Columns, and for Key Column for Network we select the option Query Term. Please make sure that the key symbol is at the gene name column, so Cytoscape will use this column to connect the node table and our imported metadata. And in the end, we click on the OK button. We can see on the table panel that our data has appeared in the node table. Now we can use our transcriptomic correlation data to color the protein-protein network nodes. For this, click on the Style button on the network panel. Let's stick with the string default style and click on the Fill Color option. From the column drop-down menu, you can select which variable you want for the coloring of your network. There are numerous variables obtained from the string database. But for now, we are going to use our own data and color the nodes based on the correlation coefficient. For this, select the Spearman's row from the columns. Now our network graph has changed. Cytoscape treats the mapping type as a discrete variable. But let's change it to continuous mapping and see what happens. Now our nodes are colored based on the row values of correlation. Since most of the correlation values are between 0.61 and 0.66, we can set the scale to highlight these differences between these two values. By a double click on the color scale, you can adjust the color properties of the mapping. Click on the Set Min and Max button to adjust the minimum and maximum values of the color scale. The minimum value is 0.6 by default. Set the maximum to 0.66, then click OK in both panels. Now our network has changed and the correlation differences are more visible. Let's try some more features of string. On the right panel there are some further queries you can use from string. Let's click on the Functional Enrichment button on the right panel. This way we will perform some basic enrichment analysis. A pop-up window will come up. Since we want to keep the default settings, just click OK. After a while a new table will appear on the table panel. The enrichment analysis result can be seen on the table panel button. Multiple databases have been used for the analysis, but for now we only want the gene pathology biological process information to be visualized. For this, click on the Filter button on the top left of the table panel. As you can see, you can select from several databases to filter on. Now we select Go Biological Process option and tick the Remove Redundant Terms. Then we click on the OK button. The table panel has changed and only Go Biological Process variables remained. Now we can color our nodes based on their biological functions. For this, just click on the Draw Charts button next to the Filter button. Now our nodes are colored based on their biological functions as well. Finally, we can see that although the RNA expression of these proteins correlates on the transcriptome level, the protein-protein interactions might differ in the protein level. Well, networks, especially biological networks, are complicated phenomena. Using Cytoscape could ease our job with the user-friendly interface and the nearly endless analysis possibilities. I highly recommend checking the Cytoscape tutorial materials and the user manual if you want to dive into the usage of Cytoscape a bit more. Don't hesitate to contact me if you have any questions. Thank you for your attention.